that I'm marking attendance, and you reminded me a couple of times, but I just want to make sure ki, in case it gets missed, just remind me. ठीक?
Okay,
We
sir.
have not missed any attendance as of now, right?
Yes, sir.
Good. Good. Okay. Okay, so we'll continue in the same vein. Okay, we are looking at the blotting techniques, right? Basically, blotting techniques are nothing but, you can have either a nucleic acid blotting technique or a protein blotting technique. When it is protein blotting technique, it is called as Western blot. And yesterday we saw nucleic acid hybridization. And hybridization simply happens because of complementarity. You know, you have a piece of either genomic DNA or RNA on the agarose gel, and when you use either DNA or RNA as a probe, by virtue of the fact that the probes have complementary sequence to what is on the sample, right? The sample gets transferred to either a nitrocellulose or nylon membrane, right? The probes have complementary sequence to it, and because they are complementary, they will go and bind to it. Okay, and once it binds, there are various ways by which we can look at this hybridization and how the this can be detected. There are many different techniques. Okay, one is chemiluminescence, one is colorimetric. There are people who use radioisotopes. So obviously, radioisotopes means you will be using X-ray films and you will be developing the autoradiograms. You know. So there are different techniques by which we can look at this hybridization. But by and large, most commonly used one used to be the radioisotope method. We will be looking in our next class, on how probes get generated. Okay. There are several ways by which we can generate a probe. But for today's class, we will quickly look at the overview of blotting techniques. There are certain commonalities, but there are also certain differences. And as I said yesterday, high media, which makes all these, agar and other things like LB media and all that, they have a teaching resource on Southern blot. We look at Southern blot, we look at a short this thing visual on Northern blot, and also a visual on Western blot. Okay, so let me quickly share the screen. These are short videos, so I think we'll get an overview. I will send a write-up. I will send a write-up soon. One thing that I realized is we are missing on our assignment part. Okay, I will probably get that assignment thing either today or by this thing by our next class. Okay, let me. The screen is visible.
Yes, sir.
All right, thank you. Let me put this on full screen mode.
If you are new to this channel, please press the subscribe button and click on the bell icon to get notified.
So I found
Hi
this.
friends, welcome back to.
I found this to be very useful, irrespective whether you're doing northern blot, southern blot, or western blot. The this apparatus or this setup is pretty much the same, whether irrespective of what you're using. Right? And when it comes to northern and southern blot, typically, you would use the samples are separated on agarose. Okay, let me check. Let me give me a minute. All right, there we go. So this is something that we saw yesterday, right? In a southern blot, you have genomic DNA, and then you have samples that have been digested with restriction enzymes. Okay, and you're going to run this agarose gel, and because DNA is negatively charged, it is going to go towards the positive pole, right? That's typically the thing. It doesn't matter whether it is DNA or RNA, both of them are negatively charged because of the phosphate backbone, okay? And because all of them have a negative charge, they all will migrate towards the positive pole. And so you can separate DNA and RNA based solely on the charge, right? So in the case of southern blot, you're going to use genomic DNA. In the case of a northern blot, you will be using, instead of genomic DNA, you will be using RNA, right? So genomic DNA versus RNA. In this case, we are comparing a normal cell versus a cancer cell. And in this case, you can actually see that the cancer cell has much higher concentration of this epidermal growth factor receptor. So this is a normal cell. You see, a small amount, but in a cancer cell, you see a very huge amount of those things. Similarly, the epidermal growth factor receptor, recognizes epidermal growth factor, and you can similarly see in a normal cell versus cancer cell, not only is the receptor expressed at a very high level, even the growth factor is expressed at a very high level. Okay. So northern blots are typically, as I said yesterday, are used to quantify the amount of RNA between any two samples, right? It can be between a normal cell and a cancer cell. It could be between any two different. It could be, a cell that is grown on a rich media versus a cell that is grown on a not so rich media, or it could be a cell that has been exposed to certain medicines versus a cell that has not been exposed to certain medicines, right? So it could be something like that. Okay. Okay, let's go back to the video. So when it comes to northern or southern blot, this sample itself will vary, right? If it's a southern blot, it is going to be genomic DNA. If it is a northern blot, it is going to be total RNA from the cell. Okay, you will be electrophoresing through agarose gels, and then you will be transferring it to a solid support, which can be a nitrocellulose, it can be a nylon membrane. I wish you people were here. We could have shown you those. How do they look like? They look pretty much like a piece of paper, except that their feel is little different. Okay, nitrocellulose and nylon membranes have the feel is very different. Okay, and then after you transfer it to a solid support, you would be hybridizing, or you'll be adding your probes, and then after washing, you'll be detecting it. You'll be detecting generally by colorimetric method. But as I said, in the earlier days, people used to use radioisotopes. Okay, because radioisotopes are harmful for human health, people have slowly replaced, the radioisotopes with colorimetric techniques. Okay, these seem to work fairly well. Okay, not as great as radioisotope. Radioisotopes are extremely sensitive. This works fairly well, decent. Okay, when it comes to Western blot, you will be running the protein samples on SDS page, polyacrylamide gel electrophoresis. Then it doesn't go well with nitrocellulose, generally gets either onto a nylon membrane or another membrane called PVDF membrane. Okay, it could be nylon or PVDF, and then from there, you will be adding your antibodies to it. Okay, you will be washing it off, and then you will be adding your detection reagents. It could be chemiluminescence. It could again be anything. It could be colorimetric. It could be even your antibodies can be tagged with radioisotopes. So you can do radioisotopic signal also. But typically, if you look at it, this is a blotting apparatus, right? You will have a sample and a gel, and then you will have either it could be your PVDF membrane or nylon membrane if it's a Western blot, or it could be nitrocellulose or nylon membrane if it is a southern or a northern blot. Okay, the rest of the apparatus remains more or less same. Okay, all the migration is from negative pole towards the positive pole. Okay, typically what happens is even with proteins, because you're running it on SDS page, sodium dodecyl sulfate uniformly gives a negative charge to all the proteins, so they all will migrate towards the positive pole. Okay, or anode. Okay. So I just thought that, so this is a gross overview, okay?
Channel. Today we're going to discuss about blotting techniques, southern, northern, and western blotting. If you are new to this channel, please press the subscribe button. And click on the bell icon to get notified.
This gives you some additional
Blotting
information.
is
That's why I wanted to
laboratory
introduce
method
this to you.
used for the detection of specific DNA, RNA, or protein from a mixture of protein DNA or the RNA. The southern blotting procedure was developed by Edward M. Sutton.
Can somebody call Vanshika to the team? I'm supposed to add, I forgot to add Vanshika. Can she join this team? I'm not sure. If somebody can see if she can be, added.
In the year 1975. He demonstrated that the restriction digested DNA can be separated by agarose gel electrophoresis and transferred onto a nylon membrane by capillary action. The gene of interest can then be visualized by incubating the blotted membrane with a radioactive probe specific for the gene of interest by placing it under the X-ray film. Let's see the different types of blotting. So there are three different types of blotting. One is the southern blotting, there is the northern blotting, and the western blotting. Southern blotting is for the detection of DNA, and northern blotting is for the detection of RNA, whereas western blotting is for the detection of proteins. The southern blotting was developed by Edward M. Sutton in 1975, and northern blotting for the detection of RNA was developed by George Stark and his colleagues in 1977. In 1981, Neil Burnett refined the process which George Stark developed and for the detection of proteins, and it is termed as Western blotting. In 1979, Tobin and his
So there is another technique called southwestern blotting. Okay, southwestern blotting is typically used for analysis of DNA binding proteins. Right. So there are proteins like as we have seen in our class, DNA polymerase, DNA ligase, restriction endonucleases, single strand DNA binding proteins. There are n number of proteins. Okay, that have this ability, transcription factors that have this ability to go and bind to DNA, and that can be detected by what is called as southwestern blotting. We are not going to look at that in the immediate feature, so I'm not using that, or I'm not saying anything about that, but there is another technique called southwestern blotting. It's a combination of southern and western blotting. Okay.
his colleagues also worked towards the protein blotting technique, but the name was coined in 1981 by Neil Burnett. So here you can see some of the reference papers, publications of that time. I will leave the links in the description below for your reference. Let's see the basic workflow of blotting technique. So for blotting, initial step is the sample preparation, followed by electrophoresis and transfer to a solid support membrane, and finally the detection of specific DNA, RNA, or the protein. The first step in the sample preparation is the isolation of nucleic acids, DNA or RNA for the southern and northern blotting. Once the nucleic acid is isolated, this is treated with restriction endonucleases to generate small fragments. And the restriction digested fragments are then loaded on agarose gel for the separation. Since the RNA is not stable, it should be handled with more care than the DNA. And RNase-free supplies like pipette tips and other reagents should be used to prevent the degradation of RNA. RNA sample also requires pre-treatment with denaturing formaldehyde to prevent the formation of secondary structures. Whereas in Western blotting, the samples from cell culture or tissue are lysed or homogenized in extraction buffer containing proteinase inhibitors. The cell debris is then removed by centrifugation and the total protein content is estimated. This is done to get the equal amount of protein loaded onto the SDS page. The samples for SDS page should be boiled in denaturing buffer to reduce the secondary and tertiary structure on the quaternary structure by breaking the disulfide bonds. Once the sample preparation is done, it has to be loaded on to agarose gel for the DNA and RNA and SDS page for the proteins. You can find the detailed video on agarose gel. I will leave the link in the description below. The same for the SDS page. So here we will focus only on the main points. Once the electric current is applied, due to the negative charge of the DNA and RNA, they will migrate towards the anode. The negative charge of the DNA is coming from the phosphate backbone. Typically, 0.5 to 2% gels are used for the separation. And the shorter fragments will move faster because they are able to pass through the agarose mesh very easily. The separated fragments in the gel can be visualized using ethylene bromide dye under the UV light. So that is an intercalating dye that binds to the DNA and that can be visualized under UV light. Coming to proteins, SDS page is the gel which is used to separate proteins. So the samples are denatured and loaded onto the stacking gel. So in SDS page, there are two types of gels are used. One is the stacking gel and the resolving gel. And the samples get separated solely on the basis of molecular weight. The stacking gel helps in protein stacking and which is usually a 5% gel. Stacking gel generally used is a 5% gel. And the resolving gel helps in the separation of proteins based on the molecular weight. The percentage of resolving gel is decided based on the molecular weight of protein of interest. And typically 8 to 15% of the resolving gel is used. The percentage of resolving gel used is 8 to 15%. The protein can be stained using Comasi brilliant blue or using a silver stain for the visualization after the gel run. Next is the blot transfer. After the DNA fragments or the RNA molecules or the proteins have been separated according to their size, they have to be transferred onto a solid support membrane in order to make these molecules accessible to specific detection. And the membranes used, the support membranes used are mostly nitrocellulose, nylon, diazo modified cellulose or PVDF. The binding mechanism is based on the cross-linking with single-stranded nucleic acid or strong hydrophobic and charged interaction with the protein. So that is the binding mechanism of the DNA, RNA or the protein to the support membrane. Since the DNA fragments are double-stranded, they need to be denatured by pretreatment with alkaline solution. Most commonly used alkaline solution is sodium hydroxide. There are two methods to transfer the molecules. The first method is to place the membrane on top of the gel that lies on a stack of wet filter paper, and then the pressure is applied by either suction or by or by placing another stack of dry paper towel and a weight on top of the membrane and the gel. So this method is called the capillary
ये
blot,
शायद
and the other method is
अंदर का
called as the
है
vacuum blot.
हाँ
The molecule will move from the gel onto the membrane with a flow of buffer by simple capillary action. And the second method utilizes the molecule's negative charge. This technique is called as the electroblot. There are two types of electroblot. One is based on the wet tank blot, and the other is the semi-dry blot. The next part is the detection part. So in this, there are sub-classification we can see. The membrane, once it is the proteins or the DNA, once it is it should be visualized. So let's go one by one. For the blocking of the membrane is to prevent the non-specific binding. Non-specific interaction is prevented by soaking the membrane in a solution containing high concentration of DNA. For the detection of DNA or RNA, the high concentration of DNA, like the herring or the salmon sperm DNA, is used in case of southern and northern blotting. Whereas for protein, the membrane is blocked generally using 5% non-fat dry milk or 3% of the BSA with detergent like Twin 20. The typical concentration used for this blocking is the 0.5% of the detergent like Twin 20. So once the membrane is blocked, this need to be washed to remove any excess blocking agents, and then can be proceeded to the hybridization stage. So hybridization is done with the specific RNA or DNA probe, and typically the concentration of the probes used are 10 to 2 to 10 nanogram per ml of the DNA or RNA probe, whereas in case of proteins, the primary antibody used is around 0.5 to 5 microgram per ml. So this part needs to be optimized to get the better results. So once the hybridization is done, the unreacted molecules need to be washed out. This is done typically using low concentration of detergent in a buffer, salt buffer solution with a small amount of detergent in it. Coming to the visualization, the radioactive probe or the autoradiobiography is typically used in the olden days, whereas nowadays it is not used because of the hazard associated with the radioactive material, and the non-radioactive methods are much cheap, faster, and they are much safe to handle. For the visualization, the DNA or RNA probes linked to biotin or fluorescent dyes or other reporter enzymes are widely used. The visualization can be based on the colorimetric detection or based on the chemiluminescence detection. For the colorimetric detection, alkaline phosphatase is the enzyme which is used that converts the chromogenic substrate to colored reaction product on the membrane, and that can be visualized. And in terms of chemiluminescence detection, HRP, in presence of hydrogen peroxide and luminol, is used as the reagents for visualizing. So chemiluminescence method is much more sensitive than the colorimetric detection. Hope you.
all right. So we saw a short thing on. Let's look at this from high media.
10x TBE. Add 450 milliliters molecular biology grade water, 50 ml of 10x TBE, 500 ml 1x TBE, 50 ml 1x TBE, 0.5 gram agarose, boil to dissolve. Allow solution to cool to about 55 to 60 degrees Celsius. Add 0.5 microliter of ethium bromide 10 milligram per microliter. Add 1x TBE. DNA sample.
That's typically what happens, when you pass current through the TB solution.
Get cutting under UV for electroblotting. Ten x transfer buffer. 100 ml of 10x transfer buffer. 900 ml of molecular biology grade water. 1 liter of 1x transfer buffer.
Can you call me about half an hour?
Add 1x transfer buffer.
That's a nitrocellulose membrane. or I don't know whether it's a nylon membrane. Both look pretty much the same, except when you feel, you will know the difference.
Place the filter paper upon wet foam pad. Place the gel piece containing DNA onto the filter paper. Place the nylon membrane.
Okay, so this is a nylon membrane. This step is being done to make sure there are no air bubbles. You know, air bubbles can prevent the transfer of DNA. So they have removed the air bubbles.
Put a filter paper upon nylon membrane.
So here, you know, they are using electricity to transfer the DNA from the gel to the nylon membrane. But you can also simply just place weight by capillary action. Also, it will go. Okay. You can. So this is called as electro transfer. That will be called as capillary transfer.
Electrophorese at 100 volts, 90 milliamps for 2 hours. Remove the nylon membrane after transfer.
DNA loading dye, you can see it on the nylon membrane.
Expose the membrane containing transferred DNA to UV for 20 minutes. Incubate the membrane containing DNA at 70 degrees Celsius for 30 minutes. Add 10 ml of pre-hybridization buffer. Add 1 gram of blocking powder.
What this essentially does is, you know, it goes and binds to wherever DNA is not there. It will go
Incubate
and
at
block
45
the
degrees Celsius, 70 to 90 RPM for 45 minutes. Hybridization buffer. Blocking powder. Add 10 ml of hybridization buffer.
Because this
1
blot
gram
is
of
smaller,
blocking powder.
they are using volumes and other reagents also in a smaller quantity. Okay.
1 vial of biotinylated probe for 10 minutes. Immediately place the vial on ice for 5 to 10 minutes. Add 15 microliters of probe to the hybridization buffer. Incubate at 45 degrees Celsius, 70 to 90 RPM for 16 hours.
So this step is the longest step, 16 hours. You could leave it overnight.
Wash buffer 1. Add 5 ml of 2x wash buffer 1. Add 5 ml of molecular biology grade water. Wash buffer 1 diluted. Add 10 ml of 1x wash buffer 1.
So this is to make sure your excess rope and excess of those milk fats, whatever was used for blocking,
Incubate
gets washed
at room
away.
temperature for 5 minutes with gentle rocking. Discard wash buffer 1 after third wash. 2x wash buffer 2. Add 5 ml of 2x wash buffer 2. Add 5 ml of molecular biology grade water. Pre-warm 1x wash buffer 2 prior to use.
So wash buffer 1 might have had some detergents.
1x wash
Now
buffer
you're
2.
using wash buffer 2 will not have detergent. So you're going to wash it off with wash buffer 2 and you'll be set for detection.
Incubate at room temperature for 5 minutes with gentle rocking. Add 10 ml of blocking buffer. Add 1 gram of blocking powder. Blocking buffer. Incubate at room temperature for 1 hour with gentle rocking. Discard the blocking buffer. And 9 ml of conjugate dilution buffer.
This is for
And
detection.
9 microliters of tween 20. Add 6 microliters of ceftavidin HRP conjugate. Add 9 ml of ceftavidin HRP conjugate buffer. Incubate at room temperature for 20 minutes with gentle rocking. Two X wash buffer 3. Add 5 ml of 2x wash buffer 3. Add 5 ml of molecular biology grade water. Add 10 ml of 1x wash buffer 3. Incubate at room temperature for 5 minutes with gentle rocking. Discard buffer and repeat the wash twice. Two x wash buffer 4. Add 5 ml of 2x wash buffer 4. Add 5 ml of molecular biology grade water. Add 10 ml of 1x wash buffer 4. Incubate at room temperature for 5 minutes with gentle rocking. Discard buffer and repeat the wash twice. Add 5 ml of TMB per H2O2. Swirl until a blue color band develops. Stop the reaction by placing the membrane in distilled water.
Okay, so this was a colorimetric detection, right? Okay, let's look at northern blot. So unlike southern blot, which can be run on TBE or TAE iodide, right? Because RNAs have extensive secondary structures, hybridization can be a difficult thing. So, in order to remove those secondary structures, RNAs are generally run on formaldehyde agarostar. It is not run on, it is basically same TAE or TBE, but it will have additional formaldehyde. Formaldehyde essentially denatures, so the RNA cannot get back the secondary structure. Okay, so for northern plots, we are going to use formaldehyde, additional thing. still have some small gaps. The previous one from high medium electroblot. This is by capillary, right? Again, hybridization is the longest step, overnight, 12 to 16 hours. All right. What they didn't show you was, the X-ray film needs to be taken out in a dark room. They will have a faint, light. Okay, you can take out the X-ray film, place it right under the this thing, blot, and leave it overnight again. Okay, depending on your signal, you could expose it for a shorter time. Otherwise, you can leave it overnight, develop it, and then after developing, you will know whether it is whether you need to do it for a shorter exposure or not. Okay, so these are the things on northern blot and southern blot. I actually wanted to do a quick thing on western blot. Let's see if it works. This is again a short one. That would complete the process of all these hybridization. You can start looking at generating probes.
Discover one-on-one coding. Welcome to the Novus Visual Protocol series. In this video, we'll learn how to perform all phases of a Western blot using the most common methods for this assay. Before we can start preparing the blot, we must first prepare our sample lysate. In this example, we'll prepare a protein lysate from culture cells. Here, we wash the cells twice with ice-cold PBS and add enough lysis buffer to cover the cells. The choice of lysis buffer depends largely upon the localization of your protein of interest. We scrape the cells and transfer the cell solution to a centrifuge tube placed on ice. In order to solubilize membrane-bound proteins, we will require stronger extraction detergents compared to isolating cytoplasmic proteins. In this example, we're using a standard RIPA buffer, which is a common buffer for obtaining maximum protein yield while extracting proteins from all cellular localizations. It is very important to include protease inhibitors in your lysis buffer, which will prevent degradation of your sample. Always use freshly prepared protease inhibitors, keep samples on ice, and work quickly. We lyse the cells by pipetting up and down, followed by incubation on ice for 30 minutes. Then, centrifuge the cells into a pellet. Discard the pellet and collect the supernatant. This is your lysate. Determine the total protein concentration of your sample lysate by testing a small portion of the lysate with a commercially available protein quantitation assay, such as the BCA. This will assist you in loading equal amounts of protein into your gel. Western blots are traditionally performed under reduced and denatured conditions. These conditions will allow proteins to be separated by their molecular weight rather than their native conformational shape or charge. To reduce and denature samples, dilute each in a loading buffer, such as the traditional Lamley buffer. This buffer contains beta-mercaptoethanol, or DTT, to reduce disulfide bridges between cysteines, SDS, to assist denaturing and provide a net negative charge to the proteins, glycerol, to allow the samples to sink into each well, bromophenol blue, to visualize the lysate, and an ionic buffer. Vortex each sample and incubate at 95 degrees Celsius for 5 minutes to completely denature the proteins. You are now ready to load your samples into an SDS page gel. For this next step, we will separate the individual proteins in our sample lysate based upon their molecular weight using a positive electrode to attract the negatively charged proteins. To do this, we load our previously prepared protein samples into a commercially available polyacrylamide gel. Gels are available in fixed percentages or gradients of acrylamide. The higher the acrylamide percentage, the smaller the pore size
in the lab.
matrix. Therefore, higher percentage gels are better for low-weight proteins, low percentage gels are better for large proteins, and gradient gels can be useful for proteins of all sizes due to their varying range and pore size. Prepare your gel by inserting it into the electrophoresis apparatus and filling with running buffer that is appropriate for your gel chemistry. Rinse the wells of the gel with running buffer and add buffer to the chambers. Load your samples into the wells. If you are unsure of the amount of protein to load, 10 to 30 micrograms of total protein is a suggested starting point, as well as titering the amount of sample loaded. You will also need to reserve at least one well for a pre-stained molecular weight ladder. The ladder will allow you to monitor protein separation during electrophoresis and subsequently verify protein weight in your sample during later analysis. Close the electrophoresis unit and connect it to a power supply. Most units typically run 45 to 60 minutes at 200 volts or until the loading buffer reaches the bottom of the gel. During this time, the negatively charged proteins in each sample will migrate towards the positively charged electrode, making their way through the polyacrylamide gel matrix.
So you can actually see, the markers separating very well.
In this next step, we will transfer our separated proteins out of the gel and into a solid membrane or blot. This is based upon the same principle as the previous step, in which an electric field is applied to move the negatively charged proteins towards a positive electrode. Transfer can occur under wet or semi-dry conditions. Here we will demonstrate the traditional wet transfer method. Start by removing the gel from its cassette, cutting off the top portion containing the wells. Notch the top left corner to indicate gel orientation. Float the gel in transfer buffer while preparing the transfer sandwich. To make the transfer sandwich, you will need a cassette, sponges, filter paper, the gel, and your choice of either PVDF or nitrocellulose membrane. PVDF must first be activated by soaking the membrane in methanol for 2 minutes, but other than this, the choice of nitrocellulose or PVDF membranes is a personal preference. Notch the top left corner to indicate blot orientation, and incubate membranes in transfer buffer for 10 minutes. Create a stack by placing the following components from the black negative cathode to the red positive anode. Sponge, filter paper, gel, membrane. Be careful not to touch the gel or membrane with your bare hands and use clean tweezers or spatulas instead. Touching the membrane during any phase can contaminate the blot and lead to an excessive background signal. Filter paper and sponge. Use a clean roller with each layer to gently roll out any bubbles that may be present, since bubbles will inhibit efficient protein transfer. Lock the cassette and place it in the transfer apparatus containing cold transfer buffer, ensuring that the cassette is properly positioned from negative to positive. In order to prevent heat buildup, it is beneficial to transfer with a cold pack in the apparatus or in the cold room with a spinner bar placed in the bottom of the chamber. Close the chamber and connect to a power supply. Perform the transfer according to the manufacturer's instructions, which is normally 100 volts for 30 to 120 minutes. After electrotransfer of our proteins to a membrane, we will now block the blot, apply a primary antibody specific for our protein of interest, and then a secondary antibody which will recognize the primary antibody. Start by removing the membrane from the cassette and rinsing 3 times in water. As an optional step, we can verify the proteins were transferred successfully by staining the membrane with Ponceau red. Incubate the membrane in Ponceau for 5 minutes and wash with water until the bands are clear. After verification, the blot can then be destained by continuing to wash with water or TBS tween until the dye is completely removed. We need to block all areas of the blot which do not already contain protein. This will prevent nonspecific binding of the antibodies and reduce overall background signal. Common blocking buffers include 5% non-fat dry milk, or VSA, in a TBS tween solution. However, do not use milk when probing with phospho-specific antibodies, as it can cause high background from its endogenous phosphoprotein, casein. Incubate the membrane with blocking solution for 1 hour at room temperature under slight agitation. Decant the blocking solution and wash with TBS tween for 5 minutes. We are now ready to add our antibody. Dilute the primary antibody in a blocking buffer at the concentration recommended on the datasheet, and incubate overnight at 4 degrees Celsius with gentle shaking. A recommended optional step is to also use a positive loading control antibody, which allows the user to verify equal amounts of total protein were loaded into each well and aids in troubleshooting by removing any uncertainties with the Western blot procedure. The next day, decant off the primary antibody and wash the membrane with large volumes of TBS tween and vigorous agitation 5 times for 5 minutes each. These stringent washes are extremely important for reducing non-specific background signals. After washing, dilute the secondary antibody in blocking solution and incubate the membrane for 1 hour at room temperature at the concentration recommended on the datasheet. In our example, the secondary is also conjugated to HRP for later detection. Decant secondary and wash the membrane with large volumes of TBS tween and vigorous agitation 5 times for 5 minutes each. You are now ready for the detection phase. In this final phase, we will demonstrate signal development using the most common, most sensitive, and most inexpensive detection method, the electrochemiluminescent, or ECL, reaction. This method utilizes the HRP enzyme, which was conjugated to the secondary to catalyze the ECL reaction and produce light. The light is then gathered onto X-ray film and developed, or digitized with the aid of a specialized camera sensitive enough for this application. We start by mixing equal parts ECL reagents in a 1-to-1 ratio according to the manufacturer's instructions. We will incubate the membrane for 3 to 5 minutes without agitation. After incubation, decant ECL mixture and use a laboratory wipe to wick off excess solution from the corner of the membrane. Place the membrane in a clear plastic wrap, such as a sheet protector, to prevent drying. Avoid letting the membrane completely dry out. We can now use a roller to push out any bubbles or excess solution. Immediately develop the membrane. Both film and camera systems allow you to adjust the exposure time in order to ensure a picture-perfect Western blot. Relative band densities can now be quantified with commercially available software. Proper molecular weight can also be verified by comparing band sizes to the molecular weight ladder.
Okay. So you can actually see, as the protein concentration increases, the signals also are much better, right? So that is all about Western blots, all the hybridization techniques. I'm sorry this class took a little longer, but I think it's worth it. Okay. We will look at the generation of probes from our next class. Okay. Any questions? Okay, so if there are no questions, we will, continue our class on the networks in our next. Okay.
Okay, sir.